This is Lecture 19, the chemosensory systems. So we're going to talk today about the three major chemical senses. That's olfactory or smell, gustatory, taste, and trigeminal, which has information about irritants in the environment. So this is phylogenetically the most primitive, so meaning that it evolved the longest time ago, but it's probably the least understood in many ways. And we'll see that as we go. So olfaction, first we're going to talk about the sense of smell. And so the receptors for smell are the olfactory receptor neurons, which are up here in the nasal cavity. So here's the nose, here's the nasal cavity, breathe in. Up here on this dorsal surface, the cells are in this sort of soft tissue, and then they have hairs or cilia that are coming out, and they have the receptors for odorants on them. These then project up to a structure called the olfactory bulb, and these then leave via the olfactory nerve and go into the central nervous system. And so again, here's the receptors, here's the olfactory bulb, and this is where the system is a bit unlike other sensory systems in that it doesn't go to the thalamus. So instead, it goes to these other targets. It's going to go to the piriform cortex. This serves as the primary olfactory cortex. So this would be something like an O1, analogous to like V1 or S1 for vision or somatosensory systems. But this evolved such a long time ago that it's not neocortex. So neocortex has 6 layers, but the piriform cortex is what's referred to as an archicortex, and it only has 3 layers. But it serves as, it has cortical and pyramidal neurons, and it has interneurons, so it has a number of homologies with neocortex, just has the 3 layers, and it's at the very base of the brain, which is one of the reasons that it's hard to study. So it's in this blue region here. Other targets are the olfactory tubercle, the amygdala, and the entorhinal cortex. So from the entorhinal cortex, this is the main input to the hippocampus. These other regions, the amygdala and olfactory tubercle, feed into the hypothalamus. So this is going to regulate emotion and homeostatic control like hunger. So if you smell food and become hungry, that's because of the hypothalamus. Also goes to the orbitofrontal cortex, which has to do with decision making and reward. So again, making decisions over whether you want to eat it. So the hypothalamus sort of triggers feelings of hunger. The hippocampus and entorhinal cortex are sort of reminding you what happened the last time you smelled these things, along with the amygdala. So if it was disgusting, then when you smell this, it's kind of a gross smell, but if it's delicious, then it activates the hypothalamus for feeding behaviors. So note that although the first step isn't the thalamus, the olfactory information comes into these other regions, and then part of this goes to the thalamus, which then feeds up to other cortical areas. So the thalamus is involved, it's just not the first stopping point before going to cortical areas. So, is it true that humans have poor olfactory capabilities compared to other animals? And that's true. What this graphic is showing is the number of receptor cells and the surface area. And so here's human. We have less than a rat, even though rats are, tiny. They're whatever, 20 to, I guess adults are close to a half a kilogram. So we're orders of magnitude bigger. Here's a typical dog, and here's a bloodhound. So notice that the typical dog and bloodhound have the same olfactory area, but the bloodhounds have about 4 billion of these olfactory receptor cells, whereas a typical dog has about 1 billion and humans have maybe 12 million. So dogs have, orders of magnitude more receptor cells, and this is why their olfaction is so much better than humans. Okay, but is it true that humans are poor at olfaction? That's not true. Humans do have some pretty extraordinary olfactory capabilities. Most of the time, it's subliminal, so you're not necessarily aware of it. One good example of this is that we can smell or perceive different isomers as different smells. One good example is carvone. So the R version smells like spearmint. The S isomer smells like caraway. So it's kind of a seedy thing. So those are quite a bit different. Interestingly, I don't know who decided to do this experiment, but humans can track scents like a dog. So if someone drags a stinky thing across a grassy area, and if you give a person sort of a cone so that the smell is coming from the ground, here's the track of the person is in kind of this fuchsia color. You can see that they're kind of tracking back and forth. The white line is where the stinky thing went. So humans can actually track, not nearly as well as dogs or other animals. Also, the humans can smell some odorants at the concentration of parts per billion, so it's a very dilute smell. And it's estimated that humans can smell between 4,000 and 10,000 different odors. So that's pretty big. And if you juxtapose this with like vision, we only see 3 different colors, right? Whereas the odor space is much, much larger. And that's because chemical compounds can have all sorts of functional groups. They can have isomers. They can vary in a lot of different ways, whereas light is just varying by wavelength and frequency. So this is why the olfaction system is a bit different. Again, here's examples of different molecules that smell quite a bit different. Here's ethanol. The threshold for sensing that is 2 millimolar, so that's thousandths of a molar. Here's a number of other chemical compounds, and they have radically different structures. Some have these aromatic rings on them, others don't. So by the time we get down to something that smells kind of earthy, we're down in the nanomolar. So this is parts per billion, and bell pepper is 0.01 nanomolar. So 1/100th of a billionth concentration. So that's quite dilute. One thing to appreciate: it's very difficult to estimate or anticipate what a smell is going to be based on the structure. So things that are structurally similar can smell quite a bit different. So there's not an orderly map. So we've talked about tonotopic mapping, somatotopic mapping, retinotopic mapping, where there's an ordered representation based on some feature of the input space to the encoding that you get. Well, chemical sensing is just different because the dimensionality of the different configurations of the molecules are quite big. It's possible to be anosic, which means smell blind. That can be for a few odors or for many odors. For instance, 1 in 10 people can't smell cyanide, and 1 in 100 people can't smell skunk. What this is showing is just how many correct people would get, normal subjects, if you test them on 7 different odorants. Most normally olfactory intact people can identify that there's 7 distinct different smells. People that are amnesic can only, they don't smell many, so they oftentimes will get 0 correct. And one other thing to look forward to as you get older, the olfactory discriminability goes down with age. So the ability to detect, so your thresholds and the ability to discriminate different odors goes down with age, and it goes down a pretty good amount. It also declines in Alzheimer's, Parkinson's disease, infection, other problems of that can compromise the olfactory bulb. So it can be in the olfactory bulb, but it can also be in the central nervous system. Interestingly, olfactory hallucinations are among the earliest signs of mental illnesses like schizophrenia and other illnesses that present with hallucinations. Oftentimes people smell things that are like burning electronics. It's a very unpleasant kind of a smell. But don't worry, if you have that, there's plenty of less significant or less serious neurological issues like migraines and other issues that can also produce olfactory hallucinations. So olfaction can produce a range of physiological responses. This can be, a peaceful kind of feeling on a spring day with flowers. It can induce hunger at Thanksgiving time when you smell the cooked turkey. It can also produce very repulsive sensations like the smell of a skunk. Probably a lot of you, if you've been hiking, you've smelled an outhouse. That doesn't make me want to eat a sandwich or anything. This guy down here, this molecule is called isonitrile, and it's rated as one of the worst odors in the world. And just a very brief whiff of this can induce severe vomiting. So, if someone's working with this and a little bit gets out of the fume hood, people start vomiting. So again, this is a testament to how the olfactory system feeds into the hypothalamus and other structures that regulate emotional responses, and they can be beyond cognitive control. So, if you smell something and it makes you want to retch, you don't have a whole lot of conscious control over that. Now, by the time we get to cortical representations, we can see that different odors do elicit different patterns of responses. So this is part of the cingulate cortex. So this is when you present a pleasant smell to someone. Here's an unpleasant smell. It's a little bit more posterior than this one. Interestingly, this is the area that also signals pain, which is kind of interesting. Again, responses can include salivation. So if you smell turkey or good food, gagging, vomiting, that's that isonitrile or a particularly ripe outhouse. Also other things like mother-infant bonding. So, a recent mother smelling the infant causes lactation and bonding, and vice versa. The infant's smell of the mother can recognize mothers by scent alone. There are also gender-specific responses to different smells, particularly sex-related smells, such as androgen-related compounds. So here's female smelling an androgen-related compound. The androgens are expressed more in males than in females. And you can see that there's a part of the hypothalamus. In females, it's activated, but if it's in males, it's a different part of the thalamus, right? So in females, it might elicit an attraction, whereas in males, it might evoke some threat-related responses. Okay, now let's talk about the quote-unquote pheromone system. So some animals have a second olfactory system called the vemeronasal system, and this is a distinct system. It's separate and it's parallel, and it's for detecting odorants that are specific to that animal species and also other species that are important, so like predators. So we see this in rodents and predators. This is why, this is the system that if a mouse or rat smells cat urine, it freezes over in the corner. So this is also why animals will scent mark stuff. So conspecifics, meaning other animals of the same species, can smell that and realize, there's a very dominant male in this area, or, there's a female in this area. They can smell that. It's one of the reasons that dogs like smelling everything when they go out for walks. So this is in rodents and in some predators. And again, there are specific receptors for these chemical compounds, and these chemicals are broadly referred to as pheromones, but that's kind of a loosey-goosey kind of a nomenclature. The vemeronasal system can also smell other types of things. And it's absent in primates, but there's questions remain regarding whether humans can detect pheromones, and I'll show you something in a little bit. In fact, I'll talk about it here. So the output of the primary olfactory areas, this is the olfactory bulb. Again, they go to these areas that are involved in decision making, for sex, for feeding, for predator avoidance, all those sorts of things. And because it taps into these emotional and memory systems, so the hypothalamus on the emotional side and the amygdala, and the hippocampus and the amygdala on the memory side, you can influence decisions by sense. So, this is the cat's kind of out of the bag on this one, but a trick for selling a house is to bake cookies or, bake some baked goods before prospective buyers tour it, because the thinking goes is that activates the hypothalamus and it gives you this sort of sense of this is home, right? That you're sort of bonded somehow to this place, and therefore you want to buy it, and you'll pay more money than you would otherwise. There's also a very sort of an infamous experiment that was done to test if there are odorants, if humans can use odorants in mate selection. And so what this experiment was is they had men wear a black t-shirt. So each guy got a different t-shirt. They wore it for 2 days. Then they put them in plastic bags, and then they gave these bags to women, and they asked, so, each woman they would come into the lab, they would get this stack of bags, and they'd ask them to open the bag, take a whiff, and then rate the attractiveness of the wearer. So they have no visual cues, no auditory cues, just by smelling the t-shirt. And the women overwhelmingly chose the shirts from men who had complementary immunohistocompatibility complexes. So what this means is that these were men who were exposed to germs and other pathogens that were different from the woman. What this means is that this would confer some advantage to the offspring because they would have some innate or actually acquired immune responses to a wider variety of pathogens. At least that's the theory. It's hard to explicitly test that theory. Every year, students ask me, well, what about men? We don't know, or at least I don't know. I suspect men are probably less picky. So, but the bottom line is that, if you're a guy and you get friend zoned, well, it might just be the pathogens that you were exposed to are too similar to a particular woman, and therefore you're rated less attractive as a sexual partner. So you need to get other diseases. Okay, so now let's talk about how olfaction is transduced. So here are these olfactory receptor neurons, and they live in this epithelial layer. They have these olfactory cilia that are coming out, and these are the areas that have the receptors for different odorants. They're in this mucus layer, right? Because they need ions and they need the proper extracellular solution. So if they're just waving in the air, they're not going to get that. So this is one of the primary functions of this mucus layer. You need it, and it's very thin. And this mucus comes from a gland, the structure known as Bowman's gland. So this is where the mucus is coming from. Because these cilia are exposed to the external world, they get odorants, but they also get harmful things like bacteria, pollen, allergens, other microorganisms. So to protect the cilia, what happens is that the Bowman's gland produces mucus, and it produces extra mucus when the body detects an illness. So this is why you get a runny or stuffy nose when you're sick. So it's the body trying to protect itself by producing extra mucus. And that flushes out bacteria or viruses or other things out of the nasal cavity. There's also one of the reasons why you can't smell or taste as well, because as the mucus builds up, now it's harder for the odorants to actually get to the receptors on the cilia. Now, the cilia of the olfactory receptor neurons, we're just going to call them ORNs, olfactory receptor neurons for now. They have receptors for specific odorants. So if you puff an odor onto the cilia, so first we'll say the receptors are on the cilia. So if you puff your odor here, and if the receptor recognizes the chemical structure of the thing that's the odorant, you get some response. But if you puff this up by the cell body, there's no receptors here, so you don't get much response. This response is just from diffusion of the chemical odorant. Interestingly, the odor receptor genes are the largest known family for most animals, or at least all the animal species that we know of. In humans, there's 950 genes. There's 1,200 in dogs. And this can be 3 to 5% of the overall genome. Not all of these are expressed, so it might be more likely that we have something like 400 different receptors being expressed. Now, like what we've seen in previous receptors, many of the segments are conserved across a wide variety of species. Right, so here's a little nematode, here's a Drosophila, here's mammals. And you'll notice that these dark blue regions are similar, these parts of the genes, and these are the transmembrane parts. These lighter parts have some functional groups on them. So we have a great deal of similarity in olfaction, and that's because, that's why fruit flies love bananas and we can smell bananas. So, there must be some similarity on the olfactory receptors so that we can detect the banana. Okay, now there are receptor subtypes, and these are heterogeneously distributed across the olfactory epithelium. So this is something kind of like, it's not, maybe the retina isn't a good analogy, but it's not that you have all kinds of receptors just willy-nilly expressed all across the olfactory epithelium. Instead, there's some groupings such that in some regions you have some particular types of receptors, in other regions you have other types of receptors. That's what's being shown up here. So here's olfactory receptor neurons, here's all of them. Here's one specific receptor type, and there's some blobs up here, but not on this part. Here's a different one, the M71. That's more on this sort of fringy part and not up here. So this is showing that there's a differential expression of particular receptor types across the epithelium. How does this happen? Well, I don't expect you to know this in detail, but we've already talked about G protein coupled signaling cascades, and this is just another example of that. Because we have very complex molecules, what happens is that there are receptors that recognize particular combinations of functional groups, and that kicks off a G protein. In this case, it's GOLF, with golf. The OLF is for olfaction. That activates adenylyl cyclase. This is a special one. It's adenylyl cyclase 3 that converts ATP to cyclic AMP. That lets calcium come into the cell. When calcium comes in, there are binding sites to the inside of a chloride channel that opens a chloride channel. Unlike what we saw in other regions of the central nervous system, in this case, chloride leaves the cell. This is a negatively charged ion. As it leaves the cell, it causes the membrane to depolarize. So it's a bit different than in other systems, but it kind of borrows the same kind of G protein coupled signaling. So very similar to phototransduction in the rods and cones. Okay, now there's a great variety of responses and that we can find in the ORNs. So some ORNs are responsive to multiple odors. So here's 3 different cinyl, isomyl acetate, and acetopherone. I'm not sure what these look like, but they smell differently. This is like a cinnamon smell. I'm not sure what these smell like. But one type of ORN will have a receptor that it's responsive to these different ones. Here's a different ORN that only likes the cineryl. It doesn't like these other ones. Here's a different ORN that doesn't like this first one at all, kind of likes the second one, and strongly activates to this third kind. So and these are just a few different kinds. Now these are 3 examples, and there's between 400 and 900 different varieties of these. So there's a large variety of different responses that you can get. This is showing this again. This is with different kind of mixtures. So instead of individual components, you can play the same kind of game with different mixtures. So here's an ORN that likes A, it likes D and E. Here's a different one that likes AF and EF. So, this one didn't like F, this one likes F, this one likes A and D, this one doesn't like A and D. So again, the point is that different ORNs like or will respond either to different individual components or mixtures of components. And so when you have a response to mixtures, that means that there are more than one type of receptor in that olfactory receptor neuron. So this is unlike the cones that respond only to one wavelength. There are some ORNs that respond to multiple different odorants. These ORNs are sensitive to the concentration. So here's a dilute odor, and then as you make it a little bit stronger, you get more action potentials, and when you make it very strong, you get an even stronger response. So that's intuitive, that you can tell the concentration by smelling. So it's not just whether it's there or not, but you can detect the concentration as well. That's probably not a surprise to anybody. Okay, these ORNs then are going to make synapses with neurons that are in the olfactory bulb, and the synapses occur at these specialized structures called the glomeruli. So here's kind of a zoomed out view. So this thing, the overall thing is the olfactory bulb, and this little kind of blue blobby thing is one of the glomeruli. This is zoomed in, and it's kind of this orb. And this is where the synaptic contacts are being made. And we'll see, here's maybe a better cartoon image of this. So here are the cilia of the olfactory receptor neurons that have the receptors. Here's the ORNs. They have a protruding, we'll call this an axon, that's coming out. It's making a synapse here. Here are the dendrites of cells that are located in the olfactory bulb, but they're making the contacts with the dendrites in these glomeruli. And again, what we're going to see is that there's some sort of patterning in the organization in the way that the olfactory bulb responds to different odorants, but it's complicated and we don't have a full map. So there's not an olfactotopic mapping, unlike the tonotopic mapping or the somatotopic mapping that we've seen before. Here's an example of this. So in this image, what they're showing is here's a 3-carbon length chain. So this would be propane. The dark spots are where it's activated. Here's if you add an additional carbon to the chain, C5, C6, and notice that the dot's moving a little bit. Here they're shown in colors. So you can see that the different length hydrocarbon chains activate different glomeruli. Last thing I'll mention about this is that there is a convergence onto the glomeruli, that the glomeruli will receive inputs from certain sets of olfactory receptor neurons, but it's complicated. And by the time you get to a complex stimuli like wine or coffee, rather than activating all of the glomeruli, what you see is that there's only a few of them are activated. And so this indicates that the olfactory system is using a sparse coding. So it's kind of unlike the cochlea, where when you have an input signal that has all sorts of frequencies, the basilar membrane is going berserk. When you have something complex with a whole lot of different odorants, it doesn't activate all of the glomeruli across the olfactory bulb. Instead, it's rather sparse. And so that's why we don't fully understand it. Okay, now let's shift over and talk about taste. So we're going from olfaction to taste. Taste encodes the aesthetic and nutritive aspects of food. The primary receptors are on taste buds of the tongue, but there are also other ones in other parts of the mouth, in what my 5-year-old calls the food tube. So they're in the soft palate, the larynx. It turns out there's even some receptors in the lungs. But we're going to talk about those that are on the taste buds. And the output is via cranial nerves and is going into the pons. So it's not going through the spinal cord. So even if you have a cervical break, you can still taste because it's coming in through the cranial nerves. Okay, so the input from the different cranial nerves comes into a nerve that's referred to as the solitary tract. This goes to the thalamus. So in this case, it's more like the canonical sensory systems that go from a sensory nerve to the thalamus, and then come into the central nervous system. So it comes into the thalamus, into the gustatory cortex, which is also known as the insula and parts of the frontal operculum. And from here it goes to the amygdala. The solitary tract also makes inputs to the hypothalamus. So this is why if you taste something and it's disgusting, it doesn't necessarily go through this whole pathway, but it can go right to the hypothalamus. And from here, it goes to the amygdala, goes to the hippocampus, it sort of branches out in a network pretty quickly. It ends up in the orbitofrontal cortex, which integrates information about taste and smell, also other sensory modalities like vision, to determine whether you want to eat something or not. Okay, so how does this work? How does the transduction work? Well, if we zoom in on the tongue, we'll see that there are these taste buds, which are more properly referred to as papilla. And the taste receptors are actually in these little crevices. So it's not on the surface, but it's in the crevice. And this is why you require saliva to taste something. So if your mouth is really, really dry and you try to eat something, A, it's hard to eat, but B, you don't really taste it because the molecules have to diffuse through liquid to get in here. And if it's dry, it can't, you just get crumbs stuck in here and it can't get in there, so you can't really taste it. Different taste modalities have different thresholds. So sweet and salty are high, so it takes millimolar, 10 to 20 millimolar, and that's because the body needs high quantities of these. Bitter is much lower because that's usually a sign that something is toxic. And so if you taste even a little bit of that, you kind of want to, spit it out of your mouth. And what's interesting is that, again, they're not the taste receptors for these different modalities, bitter, sour, sweet slash umami, and salty, are located on different parts of the tongue. They're not just uniformly mashed in there. Bitter is more towards the back. So if you just stick the tip of your tongue on something, you can't necessarily tell if it's bitter, but you can tell if it's sweet or umami. So umami is a Japanese word that translates as delicious, and this is a measure of amino acids. So it tells you if something, is rich in amino acids, because that's something that the body needs. So salty is kind of on the tip and on sort of the sides. Sour is very lateral, and again, bitter is more towards the back. And if we present these different tastants and we look in fMRI imaging, we see that there is kind of a mapping of sour, bitter, salty, sweet, and umami. A little bit hard to see. You can zoom in on your notes and see that. And so again, this is more like a tastotopic mapping, similar to the tonotopic mapping and the other kind of mappings that we've seen. But this categorization misses some things. There are other qualities that are important, like astringent, so like tea and cranberry. Don't activate these, but activate other types of receptors. Pungent and spicy, there are nociceptive elements that are in the mouth and on the tongue that respond to capsaicin that are important. Fatty, so the texture, and also metallic, right? So if you think about chewing on aluminum foil, that doesn't activate any of these receptors, but it certainly gives a strong sensation. Okay, let's zoom in on this taste bud, and what we'll see is that there are taste cells, and again, these have hair cells coming out of them. In this case, they're called microvilli, and they form this taste pore, and these give rise to a synapse, which then goes to this afferent neuron, so that joins up one of the cranial nerves. And these microvilli, these are where the receptors are for the different tastants. And what we'll see is that some of them are G protein coupled, just like what we saw in the olfactory system. But for salts and acids, they work on ion channels. So these are ionotropic channels. So the salts and the acids activate a channel, which then allows ions to go right across. Again, this is why you need saliva for this to work. So because this is open to the mouth. Sweet, bitter, and umami have receptors, and these are G protein coupled. We're not going to worry about all the details. The main point is that the cell depolarizes, lets calcium in, and that causes vesicular fusion, releases neurotransmitter, and causes excitation. This is just refining those points I was just making. So salts and acids are ionotropic. Bitter, sweet, and amino acids are metabotropic, and there are G proteins. And the G proteins differ between bitter, sweet, and amino acids. And so, we'll leave it there. We've seen this IP3 receptor before, but lets calcium in, causes depolarization. That's all you need to know about that. So we can hack this system, particularly the umami. So the amino acids can bind glutamate. And so if you have some cheap old low nutrition noodles, some pure carbohydrate, and you want to make it taste rich, what you can do is put glutamate in there, because that'll activate the umami receptors for amino acids, and it'll give you the sensation that, oh, there's some sort of protein in here. This is, like a nutrient-rich soup, whereas in fact it's really just, crappy carbohydrate noodles. So how is taste encoded? Well, there's separate receptors for the 5 taste categories, and these go to, again, the primary gustatory cortex, the insula, and the part of the operculum, and these are encoded in something that has some sort of a mapping, and we showed that before. This suggests what's referred to as a labeled line coding, meaning that if you put something in a scanner and you see where in the insula lights up when you give a tastant, you can estimate what that was. So it's kind of like somatosensory in that sense. So as I mentioned before, the receptors for taste sense aren't just on the taste buds, on the tongue, but they're also in the soft palate. And it was even recently discovered, right? So 2010, 10 years ago, it was discovered that there's even some in the lungs. And so this is why things like menthol, you can kind of feel it in your lungs, and that's because there are receptors there. Okay, taste and smell interact. This is why, when you have a bad cold and you're stuffed up, your olfaction suffers because now you have a lot of mucus and the odorants can't get to the ORNs. When you deprive that information, then things can taste differently. Interestingly, some people have a heightened sense of taste. They have some receptors that other people don't have, and these people are called super tasters, and you can buy these little kits to test. So you put this little piece of paper on, and it has a ligand for these receptors that only some people have. And some people, it'll taste like something to some people, it'll taste like just paper to other people. These people and others often perceive things like broccoli to be very bitter. So if you have a kid, like my kid, I told my kid about this one time, and she said, it's unfair to give me broccoli because maybe I'm a super taster and it tastes worse to me than it tastes to you, so it's unfair to make me eat it. So that was a pretty solid argument, but in the end I won. Okay, the third system, so we talked about olfactory, gustatory. The third system we're going to talk about is called the trigeminal chemosensory system, and this is all about detecting threats or insults to your sensitive organs that are in your face. So your eyes, ears, mouth, these are all very sensitive, and if you have some sort of pollutant in there, it could be pollen, could be smog, could be pollen, something like that, ammonia, ethanol. So if that comes in, what happens is that you start to produce tears and mucus to try to flush all that stuff out. So these nociceptive neurons are located in the face, the cornea of the eyes, and in the oral and facial cavities. So this trigeminal system, it's more, it's in, it's on the cornea, so it's a little bit broader than just the taste and olfactory systems. And this is the main basis of like pepper spray. So if you get pepper sprayed in the face, you get uncontrolled salivation, tearing, nasal secretion, sweating, decreased respiratory rate, because you don't want to be breathing in the toxicants, right? So, you can't avoid but turn away. You're very much incapacitated. Okay, and here are the key concepts for this lecture, and I will see you on the next one.